Hello, my name is Ling Wen, and today I would like to present to you the influenza disease, aka flu, and its genetic evolution. Let's start with the flu in 1918 that killed at least 50 million people worldwide. It's also called the Spanish flu, although many suggest that it was most likely originate from Kansas in spring 1918. It had two outstanding characteristics. One, it killed millions of people, and two, most of them were in the prime of their life. It infected one-third of the world's population, and the pandemic came in three waves. The first wave was in spring 1918 when the US was engaged in World War I. Second wave was in fall 1918, the most fatal one. Many health professionals served in the US military during World War I, resulting in shortage of medical personnel around the US. It got so bad that public gatherings were prohibited. And it ended with the third wave in winter 1918. The Spanish flu also left an extensive impact on the genetic evolution of influenza virus and also called the mother of all influenza pandemics. Influenza A pandemic since that time, and indeed almost all cases of influenza A worldwide have been caused by descendants of 1918 virus, including drifted H1N1 viruses and resorted H2N2, H3N2 viruses. These viruses have been circulating worldwide for 29 years and showed little evidence of extinction. So what is influenza? Influenza viruses are among the most common causes of human respiratory infections and among the most significant because they cause high morbidity and mortality. Influenza outbreaks have apparently occurred since at least the Middle Age or ancient time. In the United States, influenza results in approximately 200,000 hospitalizations and 36,000 deaths in a typical endemic season. Influenza viruses can cause serious infections and leading to development of pneumonia. Influenza virus infection includes both upper and lower respiratory tract involvement. Death may proceed in as little as 48 hours after onset of symptoms. People with chronic pulmonary or cardiac disease or diabetes are at high risk of developing severe complications from influenza A viruses. These influenza viruses came from the same family, Orthomyxoviridae is characterized by segmented negative strand of RNA genomes. There are three types, A, B, and C. These are the characteristics that cover all of the influenza viruses, but not all three viruses have all six characteristics, except for A. Influenza type A and B are able to cause flu epidemics. These are the epidemics is basically the elevation in cases during some months of the year, right? The highest rate of flu disease in a year is around winter time. Influenza type C does not cause epidemics because it has low level activity throughout the year. There are currently vaccines for A, B, but not C. Nearly all adults have been affected with influenza C virus because it causes mild upper respiratory tract illness. Influenza virus C is not as severe as the other two. Genetic drift and shift. Genetic drift occurs in influenza virus A and B. Shift occurs in influenza virus A only, which is the reason to many pandemics. Shifts and drifts are two genetic changes in the virus genomes. We will discuss about these two more in depth later. Influenza A affects birds, pigs, horses, dogs, and cats. Influenza C affects pigs and dogs, while influenza B does not affect animals. All three infect humans. Understanding the structure will help us learn about their functions. All three types of these viruses are enveloped viruses. Influenza A virus has 3 membrane proteins. Influenza B virus has 4 proteins in the envelope. Both A and B have hemagglutinin, abbreviated as HA, and neuraminidase, abbreviated as NA proteins. Influenza B viruses cause the same spectrum of disease as influenza A. However, influenza B viruses do not cause pandemics. Influenza C virus has the major envelope glycoprotein, which is HEF, hemagglutinin esterase fusion, having the functions of both HA and NA. And this is why it does not have genetic shift or drift. The C also contains 7 RNA segments and not 8 RNA segments like A and B. So how do the virions attack the host cells? These 6 steps that I have here on the slide is a general guideline or process of infections that virologists lay out. Step 1 is attachment, 2 entry, 3 translation of mRNA into protein, 4 is genome replication. In the case of influenza viruses, they only produce more RNA and not DNA because they only have RNA. 5 is assembly of new particles, 6 release particles from the cell. So what really the process looks like is that when virus enters into the respiratory tract, they are trapped in mucus. And the hemagglutinin protein from the virus attaches to the cell receptor, aka sialic acid, and initiates the process of virus entry into the cells. The spikes that you see here on the virus are HA protein, and the receptor is sialic acid, a small sugar that is attached to the protein. When a sugar binds with a protein, it's called glycoprotein. Virus is then endocytosed into the endosome. And then the RNA from virus get into the cell and replicates themselves. Viral proteins and viral RNA are made. The cell is taken over, and then the viral RNA turns the cell into a factory of viral production. Now the virus is seeking out and try to attack other target cells using its hemagglutinin protein. They have to get loose somehow, right? But the HA protein is still attached to the sialic acid. Here, the NA proteins, the neuraminidase, cuts the sialic acid and break the HA free or the virus free. The cell that is already infected that they just leave, that cell starts getting damaged and eventually dies. And that this whole process creates inflammation and causes the immune system to run wild. So why do people call the 1918 influenza viruses the mother of all pandemic? Why do we need to get vaccinated every year? This is because of genetic shift and drift of the virus. The segmented genomes and the error-prone RNA-dependent RNA polymerase enable these viruses to undergo antigenic shift and drift. It helps the viruses to evade the adaptive immune system. Antigenic drift occurs in both influenza type A and B viruses. These are the minor changes over time by point mutation in gene. Antigenic changes over time as virus replicates. Antigenic shift is seen in influenza A virus. They cause abrupt major changes which create new subtypes. This happens when 2 viruses attack the same cell. Once the cell is taken over and becomes a viral RNA factory, it produces viruses that are either similar or different from the parent viruses. The ones that are different and have a mix of RNA from the 2 initial viruses is caused this is caused by exchange of gene segments, resulting in new hemagglutinin or neuraminidase protein in influenza viruses that infect humans. There are 17 types of HA and 10 types of NA proteins. An influenza virus needs some HA and NA, so we have to multiply 17 by 10 to get the total of all combinations, which yields 170 types. For example, if we have the 5th type of HA and 1st type of NA combined together, we have H5N1. And this is the cause to so many pandemics and the reason why many viral descendants from the 1918 influenza pandemic can get away from antibodies already created by vaccine. Since viruses constantly undergo genetic shift and drift, antibodies produced by our bodies or given by vaccines cannot keep up. Humans keep getting sick and dying from influenza viruses when the viruses can be as old as the earth. It is very hard to predict what strain of virus encounters the next pandemic. Therefore, we always have to be cautious and do everything to prevent and prepare for the next pandemic. Thank you for listening.